Oh, hey, I didn't see you there. Welcome back to this edition of Pandemic Radio. On this week's episode, we'll be discussing the always exciting subfield of neuroepidemiology. So, today we're going to put the neuro in epidemiology. And I suppose we should begin our discussion with a little definition. Neuroepidemiology. Hopefully, at this point, you are all relatively familiar with epidemiology and what that means, the study of disease in human populations. We are just adding neuro. So, we're essentially looking at neurological diseases and applying all the epidemiological principles that we've learned so far. So, think about disease incidence and prevalence and case control studies, randomized control studies, etc., applied specifically to the unique challenges of neurologic diseases. Now, this is in many ways an emerging and largely, um, I would say, new subfield, looking at brain health and studying brain health using the tools of epidemiology. Um, the health of brain systems has been called, quote, the greatest challenge of societies in the 21st century. And this was from the editor in chief of the Lancet Neurology, Dr. Elena Becker Barrasso, and I would argue is probably a little biased since she is the editor in chief of a neurology journal. But I would tend to agree that um, if not the greatest challenge, understanding brain health and brain function is a significant challenge for the medical community and for epidemiologists. So let's continue our little exploration into some of these terms. Neurological disorders or neurological diseases are those of the central and peripheral nervous system. So this includes diseases of the brain, the spinal cord, and the nerves that connect them. So really, it's more than just the brain. When you think of neuroepidemiology, you probably think first of the brain, but it's anything in the nervous system, including the spinal cord, cranial nerves, peripheral nerves, nerve roots, and so forth. So this is really diseases, morbidity and mortality associated with this system is what we're talking about when we're looking at neurological or neuroepidemiology. And this is a really big subfield. So epidemiology, as a whole is quite large, and then we have all these branches underneath the umbrella of epidemiology. Neuroepidemiology is quite large because there's a lot of neurologic diseases. Over 600 neurologic diseases have been identified, and they constitute a significant source of death and disability worldwide. So when you put them all together and you compare them to all the other diseases on the planet that affect humans, they are the second leading cause of death after heart disease, and they're actually the leading cause of disability, which I think is a probably surprising statistic or fact for a lot of you. So mortality second, morbidity first. So, the global burden of neurological disorders have been further classified into 15 distinct disorder categories. So, tetanus, meningitis, encephalitis, stroke. And one we're going to focus on today in this talk is Alzheimer's disease. But neurologic disorders can also include things like migraines and headaches. And I would argue that who hasn't, or I would ask who hasn't had a headache or migraine or known someone who suffered from a migraine. And you can start to understand why uh, this group of disorders is so common and such a major contributor to disability worldwide, is because some of these disorders are quite common. So the four largest contributors of neurological DALYs, and again, DALYs, disability adjusted life years. So if you are diagnosed with a disease or you die from a disease, that takes away healthy time that you would otherwise be living a healthy life. So if you are expected to live until 70, let's say that the average life expectancy is 70, and you are diagnosed with Alzheimer's when you're 60, and that basically reduces your kind of health by half. You've um, lost uh, 5 years of healthy life. If you die when you're 60, you've lost 10 years because 70 minus 60. So the top four contributors of DALYs, the things that cause sickness and death worldwide: stroke, migraines, Alzheimer's and other dementias, and meningitis. So this is globally the top contributors of DALYs. And then the burden of disease studies also tend to break these disorders down by geographic location. And so you can see where we are in what's called high income North America. The distribution of disorders in terms of DALYs differs a little bit. So the top three in North America remain stroke, migraine, and Alzheimer's disease. But number four is actually spinal cord injury. And you can see if you do a comparison with other countries, you do start to see some differences emerge. But overall, stroke, migraine, and Alzheimer's are really kind of at the top, depending, regardless of where you are in the world. So, in addition to there being geographic differences in neurologic disorders, there is also a sex difference between males and females. So, age standardized DALYs are actually significantly higher in males compared to females. And the male to female ratio is actually 1.12. So not tremendously higher, but statistically significant that males are more likely to suffer morbidity and mortality from neurologic diseases compared to females. And let's go over a few Snapple facts. Why not? I mean, that information was slightly depressing, and I don't know about you, but what cheers me up more than a good neuroepi Snapple fact? So let's start with let's kind of warm our brains up a little with just kind of a random Snapple fact, and I like this one. And my apologies if this is going to keep you up at night or if you have arachnophobia, but I found this to be interesting. The average human eats 8 spiders in his or her lifetime while sleeping, and I'm not going to cite that because I have no idea if that's true. I just have heard that, and I'm going to put it there. But let's go on to what we do know is true and what I can cite: that approximately 1 in 6 women and 1 in 10 men on the planet experienced a migraine in 2016. And this is from the Global Burden of Disease study. So you can see, very, very common. Migraine is especially burdensome among women aged 15 to 49. So you've probably either had migraines, suffered from them, or known someone who has, because it's such a common condition. Parkinson's disease is the fastest growing neurological disorder that we know about. Interesting. So here is one of the things that makes neuroepidemiology challenging, and I would argue a little frustrating. Is that so? We know that these disorders are significant contributors to morbidity and mortality and health problems. And what do we do in epidemiology? We ask why. We ask, why do those conditions exist? Why do migraines affect that group of people compared to this group of people? We want to look for risk factors. We want to look for the causes of these health outcomes. And so neuroepidemiologists have been looking for a variety of different risk factors to try to explain why do neurological diseases contribute to such a high proportion of the burden of disease? And because they're epidemiologists at the end of the day, they are looking for associations. They're asking why. And here's the kicker: very few causative explanations have been found. So, this was a review study looking at 84 different risk factors that were quantified, looking at all of these possible explanations for neurological disorders. And it's very difficult in neuroepidemiology to find significant risk factors. So, remember your odds ratios and your relative risks. If they're above one, they increase the likelihood of the outcome. It's really hard to find large measures of association in neurological epidemiology. We just don't know a lot of the times what is causing or what specific combination of risk factors are causing neurological disorders. But one thing that neuroepidemiologists are looking for increasingly is a geographic explanation. So, where you live could determine the risk for example, Alzheimer's disease. This is a map of age standardized prevalence of Alzheimer's disease. And you guys all remember the glorious benefits of age standardizing your prevalence rates, right? That's right. I'm proud of you guys. So we know that we can look at a map like this and we are confident, one, that we've adjusted for size differences in the population, but also for age differences in these different countries. And after adjusting those two things, because we know that, especially for a disease like Alzheimer's, age and population size are going to be big risk factors. And we know that age is a particularly big risk factor for a disease like Alzheimer's, right? So when we age standardize a statistic like this, we basically have controlled for differences in age and the size of the population. And when you see this data mapped, you can see that there are non-random kind of patterns. And when I look at a map like this, I look especially at, like, parts of Northern Africa, the Saudi Arabian Peninsula, and even down in South America, in Brazil, the risk seems to be a lot higher than other places in the world. And as a good epidemiologist, you should all be asking, why? So I want to talk about Alzheimer's specifically and use this as our first case study in looking at how neuroepidemiology can be useful in understanding this disease. So, Alzheimer's disease is a type of dementia. It is the most common form of dementia in the United States, and it is characterized by progressive memory loss, a general decline in cognitive skills, which ultimately lead to adverse behavioral changes. The average age at death when someone is diagnosed with Alzheimer's disease and ultimately dies as a result, the average age is approximately 85. Um, Alzheimer's is not a normal part of aging, so you don't just get Alzheimer's just as a fact, because you're, getting older, but it is a significant risk factor. And a majority of people who are diagnosed with Alzheimer's disease are over 65. Here, women are at actually an increased risk of death compared to men. And the World Health Organization estimates that by 2030, and that's not very far away, about 66 million people in the world will have the disease. So this is a growing public health problem. And we're going to talk a little bit today on Pandemic Radio why neuroepidemiology can be useful in understanding this problem. So I mentioned a few slides ago that, well, we've looked at all these different risk factors and it's really hard to figure out or pinpoint what is causing neurological disorders. And Alzheimer's is no different. We really don't know what factors, either lifestyle, environment, genetic, etc. We really don't know what combination of factors increases or changes risk of Alzheimer's. And because of that, thus far, we really don't have any good treatment for this disease. Alzheimer's has resisted attempts to treat it, and part of that is because we really just don't have a good grasp of what is even causing it. The medication that we do have, the current drugs that we have to treat it, are basically for people who are already showing severe signs of the disease, who are already cognitively impaired. And at this point, it's really not that beneficial because when someone with Alzheimer's is showing behavioral changes, the brain has already been damaged. And when the brain is damaged, there's no really kind of undoing it. So the uh, one of the prominent Alzheimer's researchers in the world, Dr. John C. Morris at Washington University in St. Louis. Hey, I taught there for a little while. It's kind of fun. He says that perhaps the reason our therapies have been ineffective or mostly ineffective is that we're administering them too late. Okay. So the changes to your brain physiology may be slow, and maybe by the time we catch it, it's too late. Maybe by the time you start showing symptoms, your brain has already been damaged past the point of really any kind of significant help. So the current drugs that we use to help with Alzheimer's disease um, don't really do that much relative to other kinds of drugs and other health conditions. So, in many ways, Alzheimer's remains a mystery disease. We really don't know what starts it. We really don't know that much about the risk factors and the kind of quote, why though, right? We're epidemiologists and we're all about that why though. Here's what we do know: people with Alzheimer's disease develop two things. They develop what are called amyloid plaques, which are essentially clusters of abnormal protein deposits between neurons. And you can see on the figure at the bottom to the right, you see those little dark kind of clusters are basically little deposits of this abnormally folded protein called amyloid plaques. The other telltale sign of brain anatomy of people who have Alzheimer's is this thing called neurofibrillary tangles. And this is essentially occurring inside of the neurons. This is the, your brain cells. And these are like these twisted strands of protein called tau. Uh, both of these together essentially wreak physical havoc on your brain. And when you autopsy someone with Alzheimer's disease and you compare the brain of someone with Alzheimer's to a healthy, normal brain, you see a pretty substantial difference. You can really tell an Alzheimer's afflicted brain from a normal, healthy brain that's aged normally. And it's thought that it's these presence of neurofibrillary tangles and amyloid plaques that are the main culprit here, because we don't see these things in normal, healthy brains. We do see these things in Alzheimer's afflicted brains. And the severity and ultimate outcome of Alzheimer's disease is in many ways influenced by the presence of these amyloid plaques and tau tangles. So, people who tend to be diagnosed with Alzheimer's earlier in life, early onset Alzheimer's, uh, tend to have amyloid plaques at a higher rate earlier on, and this is kind of a telltale sign. But tau tangles are also present, so we have these two physical manifestations in the brain, these plaques and these tangles, um, and the medical community has really been looking at these things as like the culprits. And so if these are the kind of culprits, we want to develop medications to try to prevent them from forming in the brain. So the goal has been and continues to be that we need to do something about the disease earlier. But preventive research is difficult, right? By the time someone is diagnosed with Alzheimer's, again, it's usually probably a little too late because the damage has already begun. These plaques and these tangles have already started to form, and the brain has already been damaged. What you really need to do is try to catch people before this damage occurs so that you can alter the development of plaque and tangles before they cause damage. But this is difficult because how do you diagnose someone with Alzheimer's disease before they develop symptoms? How do you know that someone is going to get Alzheimer's disease, but catch them before these plaques and these tangles cause damage to the brain? This is what makes neuroepidemiology of Alzheimer's disease so difficult. Because the average age is greater than 65. So, any kind of prevention trials, any kind of randomized controlled trials that are going to follow people over long periods of time, it's difficult. There's a lot of logistic problems there. This is too long of a wait for people to develop Alzheimer's. Because if you enroll people in the study when they're in their 70s or early 80s, that's obviously problematic. Okay, you don't have the luxury of following people for that long. Until this rather unusual cohort was discovered. And this is, I think, one of the coolest cohorts of any epidemiology story I've ever read or come across. And that's saying something because I'm an infectious disease epidemiologist, right? That's my jam. Okay. Neuroepidemiology is cool and all, but for me, it's all about those vector borne diseases, right? Even for someone like me, this cohort is very exciting. Okay. So I'm going to tell you a little bit about this group of people. Okay. Cohorts, AKA group of people. Okay. This cohort exists in a little town north of Medellin in Colombia, South America. There's a little map for you right there. Oop, where's my arrow? Well, there it is. Okay. This location is unique because a majority of the people who develop Alzheimer's disease in this little town get it early. The average age is 40 and full blown Alzheimer's by 47. Okay, that's very unusual to have a region where essentially everyone who's getting Alzheimer's disease is in their early 40s. Most people in this town share a common bloodline. It's essentially a large extended family. And it also happens to be the world's largest extended family to experience Alzheimer's disease. There's actually a genetic component to this group, and because they all share a distant shared bloodline, this group, more than any other group on the planet, is at increased risk for Alzheimer's disease. And not only for increased risk of Alzheimer's disease, but early onset Alzheimer's disease. So obviously, this becomes a pretty tempting cohort to study, and neuroepidemiologists have um, been looking at this group as a potential game changer for understanding Alzheimer's and treating Alzheimer's. There are some logistical challenges, just like with conducting any epidemiology research, but some that are unique to this area. Medellin is colloquially called the murder capital of the world, so it's a dangerous area or can be dangerous. Overall, low socioeconomic status and education levels. And typically, when you have regions with lower education levels, you have um, more challenges in terms of people buying into the research and participating. And there are a lot of misconceptions about Alzheimer's disease in this community. A lot of misinformation and myths and so forth. So some of the common misconceptions about Alzheimer's are that it's transmitted sexually, um, and people can become infected with sex from a quote already mad woman. This is a common. They actually interviewed people, and this was a common response that people said in this area. That it could be transmitted through blood of a sufferer's, wounds, and there's a lot of stigma about this disease in the community. And because of this, it does make research, difficult when people don't really have a clear grasp or understanding of what's going on. So let's get down to the nuts and bolts here. Why is this group so special, and why is this potentially a game changer in neuroepidemiology? It has to do with this little mutation. So we know that, genes, DNA, they basically are blueprints for, creating protein, different proteins for doing different stuff. And when your genetic material gets copied over and over and over, sometimes there's a copy mistake, right? If you copy like a large stack of paper, there's a chance that maybe like one of those copies gets smudged or something, right? And this is a mutation. There is a particular mutation that if the person has it, they will develop Alzheimer's disease. PISA will guarantee Alzheimer's. So let that just sink in for a second. This mutation globally is pretty rare. And this mutation is present in about 2% of the world's Alzheimer's population. In our cohort, 30%. Okay, so what does this mean? Because this group in this little town in South America share a common bloodline, meaning they're all pretty much distantly related. There wasn't a lot of moving in and out of the community, so there's this kind of shared genetic background in this group. So not only do they have this kind of shared genetic background, that when you have a rare mutation like this, it's more likely to be amplified in a population like this. So this group in South America has really high rates of this rare genetic mutation that we know if this mutation exists, we know that the person will get Alzheimer's. So imagine being tested for this mutated gene, because you can test for it, and not just, well, you're likely to or whatever, it's you're going to get early onset Alzheimer's disease. So this mutation has been kind of concentrated and passed down, and has been a common inherited mutation that guarantees Alzheimer's. Okay. And this occurs because we basically have this genetic bottleneck in this area where large families and intermarriage have basically accelerated the prevalence of this mutation, has made this mutation more common in this cohort than other parts of the world. This is potentially groundbreaking for studying Alzheimer's, because it opens up the possibility that we could treat the disease before it starts. And if we can catch it early enough, we could actually develop therapies to prevent Alzheimer's disease. Because remember what I said at the beginning, one of the challenges is uh, in treating Alzheimer's disease is by the time we catch it, it's too late. By the time symptoms emerge, it's already too late and the damage has been done. But if we can find people with this gene early enough, we know that they are going to get Alzheimer's disease. Okay. The problem with that is we don't have a treatment yet. So we can tell someone that they have this genetic mutation and we can say, okay, you have it, but we can't do anything for you. All we can do is identify people who we know are going to get Alzheimer's disease. So cue this study. And this is what I mean by, I think, being one of the coolest epidemiology studies I have ever come across, because of this cohort and because of the potential implications for Alzheimer's disease. So I'm going to talk about this randomized control trial that was done with this group. This team of researchers recruited 300 residents of this community. They were all over the age of 30, and none of them have developed symptoms of Alzheimer's disease yet. So they were all young, healthy residents of this community. They tested everyone in this group for this mutation, and 1/3 of them tested positive. They decided to test this medication that's an experimental drug that blocks plaque from forming in the brain. And remember what I said, that Alzheimer's disease brains have plaque formations and tangles. The theory is that if we can give someone medication that blocks the formation of this plaque early enough, we can actually stall or prevent or slow the disease from happening. The remaining 200 people, so 100 of the people tested positive. The remaining 200 participants receive a placebo. So let me kind of walk through this. Okay, so let's start with our group of 300. Citizens from this area, and we're going to test all of their blood, and we're going to find who has this rare genetic mutation that guarantees Alzheimer's and who doesn't. And we find that we can kind of group people by whether they have it and they don't, but they don't kick the people out. So this bottom box here, some of the people don't have this mutation, meaning they are not going to get Alzheimer's disease. But we don't exclude them from the study because at this point, the researchers are not telling anyone whether they have this mutation or not, because at this point, there's nothing we can do. So everyone enrolled in this study, nobody knows whether they have the mutation or not at this point. It's double blinded. Okay. Of those who have the mutation, 1/3 will get the experimental drug that is thought to block plaque formation, and the other group will get a placebo. Follow them over time, and the goal is to say who develops Alzheimer's. We know that these groups in red, those with the mutation, will develop Alzheimer's, 100%. So what they're looking for is if this new drug, if this experimental drug does anything, that you would expect to see, like after 5 years, you would expect there to be a lot less Alzheimer's disease in this top group. If this drug that blocks plaque formation is ineffective and doesn't do anything, you would expect there to be equal amounts of Alzheimer's disease in these groups. This is important either way. If you notice at the end of this study that both groups have the same amount of Alzheimer's disease, it means that blocking plaque early is not the way to go. And maybe we should be focusing on those tangles I talked about earlier and not the plaque. Okay. If we do see a significant difference in Alzheimer's between these groups, it suggests that a drug to block plaque might be the cure to Alzheimer's disease. So, either way, we're learning from this group a ton about Alzheimer's potentially. And what's exciting about this is an ongoing research study. And we're right about here in a lot of these trials. And a lot of the results are either coming in right now or will be coming in the next year or so from these kinds of trials, which is pretty exciting. So I posted some films on the course website that do a really good job, I think, of explaining this case study in a little more detail. And I encourage you to watch these videos, and together with this lecture, I hope gives you a pretty clear idea of how um, cool this case study is. And even if you're not a neuroepidemiology type of person like yours truly, this is still really cool. So enjoy these films. And I would encourage you guys, if you want to kill some time in your quarantine and you want to poke around for any of these trials, randomized control trials that have been done out there and are starting to show results, please let me know. Send them my way. I'd love to know a little bit more about it. But that's neuroepidemiology part one, looking at Alzheimer's disease and a rather unique cohort in helping us understand a little bit more about this disease. All right, wash your hands, wear your masks, physically isolate, but don't socially isolate. Call your mom, call your dad, call your cousin, pet your dog, and I'll see you next time.